Welcome to Prudence Coaching students. Today we are going to learn about the unit 2 portion of biology, that is plant physiology or absorption of roots. So let's start about this chapter. We will learn chapter wise SAQ so that it helps you a lot in your exam, which is knocking at the door as well. So let's learn about this. First of all, we need to know what is plant physiology. Plant physiology is the branch of biology which deals with the life functions of the plant. It includes the functioning of cells, tissues, organs and organ systems and the organisms as a whole. In this chapter we are basically going to learn about its application and the several process like osmosis, diffusion, active transport, hypotonic, isotonic, and hypertonic solutions, and so on, and its application. So let's learn about this. The first question is, what are the major characteristics of roots for absorbing water? The ability of roots to draw water from the soil is dependent on three major characteristics. Number one, a huge surface area provided by rootlets and the root hairs. Number two, root hairs containing the solution which is known as cells up at a concentration higher than that of the surrounding soil water, and root hairs having thin walls. So these are the three major characteristics of the roots for absorbing water. Now we have shown here the picture of a freshly germinated seed showing the single root with its root hairs. So what is the root hair? Root hair is small hair-like structure which that comes out from the central root. This is the central root portion, and these things, these black hair-like substances are the root hairs which helps a central root for absorbing water. Now we'll move to the next one. This is the full grown root hair. You can see the full grown root hair. This is the picture of the full grown root hair, and here this portion is being known as the full grown root hair, and within this, these are the parts of this full grown root hair. This is epidermal cell. The outside portion is known as cell wall. Inside the cell wall there is cell membrane, and inside the cell membrane there is cytoplasm. And vacuole, especially in plants, the size of the vacuole are larger than the human or the animal cell. And the most important part of a cell is nucleus, which is red in color, which is mentioned in here. So that's all about this full grown root hair. You need to know about all these points because in exam this pointing is very much important for you. Now we'll know about imbibition. What is imbibition? Imbibition is a phenomenon by which the living or dead plant cells absorb water by surface attraction. Substances which are made up of cellulose or proteins are hydrophilic. Hydrophilic means strong affinity for water. That means those who are hydrophilic in nature, they have strong affinity for water. They imbibe water or moisture and swell up. As a result, dry seeds, wooden doors swell up on contact with water or on exposure to the moisture. Due to the imbibitional pressure, the seeds walls ruptures in case of germinating seeds. It is also an important force in the ascent of sap. So what is the application of imbibition? The process of imbibition is the swelling up of seeds and the wooden doors which swells up due to absorbing water, and this process is completely dependent on imbibition, and it also helps in creating the important force of ascent of sap as well. Next is diffusion. Diffusion is very much important for your exam, so you need to give strength on this point. Diffusion is the free movement of molecules of a substance. This substance can be either liquid, gas, or solid. No, not solid. This diffusion occurs only in liquid or gas. So, diffusion is the free movement of molecules from the region of their higher concentration to the region of their lower concentration when the two are in a direct contact. So, in diffusion, the two main points are essential or most important, that is The movement of the molecule occurs from the region of higher concentration to the region of lower concentration, and in diffusion the two substances must be in direct contact. So that's all about diffusion. Now this picture clearly depicts the picture of diffusion. Here this red colored thing is soluble dye in water. So this soluble dye is being spreading from its higher region to that of the lower region. So we can see in B, or in funnel B, there is the dye has been spreaded already inside the water, and this is spreading from its higher region to that of the lower region. So we can see this picture and clearly depict that this is the process of diffusion, and in C this things has been completely mixed up. So this whole things is the experiment of a diffusion. Now we'll know about osmosis. Osmosis is the movement of water molecules from the region of higher concentration to the region of lower concentration through a semi-permeable membrane. So we can see that osmosis is quite alike to that of diffusion, but in osmosis there is the movement occurs only in the water molecules, and this process is which is alike diffusion. Here we need to use the semi-permeable membrane. Without semi-permeable membrane this osmosis is impossible. Now here the picture or the experiment of osmosis has been mentioned here. We can see this within this thistle funnel there is concentrated sugar solution, and this cellophane paper which is used on the surface of the thistle funnel, this is working here as a semi-permeable membrane, and outside the thistle funnel there is a normal funnel where water is being kept. And after some time, when this thistle funnel will be suspended inside this water containing funnel, will find that the water level in this thistle funnel will rise up after some time due to the osmotic pressure, or as there will be the process of osmosis occurring, then there the water level will be risen up after some time. So that is the process of osmosis. With reference to a cell, the osmosis can be either inward or outward depending on the extent of the concentration of the solution surrounding it. So there are mainly two types of osmosis. They are endosmosis. Endo means inward, around osmo means push or thrust. So inward thrust and outward thrust or exosmosis. First of all, we need to know about endosmosis, and this is the most important point, that apart from its definition, its application is most important for your exam. Endosmosis is the inward diffusion of water through a semi-permeable membrane when the surrounding solution is less concentrated. This tends to swell up the cell. So the application of endosmosis is that due to endosmosis a cell swells up. Exosmosis. Exosmosis means outward thrust. So exosmosis is the outward diffusion of water through a semi-permeable membrane when the surrounding solution is more concentrated, and due to this it tends to cause shrinkage of the cell. So as we can see, due to endosmosis the cell tends to swell up, and due to exosmosis the cell shrinkage. Now here is the difference has been mentioned for diffusion, osmosis and active transport. First of all, we have to provide the definition which is mentioned here. Next, we will see the substances which are being mentioned or used in diffusion. Liquids and gases can be diffused over considerable distance, and in osmosis the water only used or take part in the transport, and in active transport cell energy from ATP is needed for transportation. Diffusion. In diffusion, the movement of the molecules is of solute or solvent, and in osmosis, movement of the molecules of only water as a solvent is required, and in active transport movement of the ions only other than water. Number 4, rapid. This diffusion occurs rapidly in gases, but it is slower in solution. And in respect to diffusion, this osmosis is a very slow process, where active transport is a rapid process. Number 5, diffusion. In diffusion, transport from high to low region occurs as a gradient, and in osmosis, transport from a solution of low to high concentration occurs. And in active transport, it is a movement of molecules against the concentrated concentration gradient. Number 6, diffusion occurs with or without a non-living permeable membrane, but in osmosis, semi-permeable membrane is used. And in active transport, a living selective membrane is essential. So that's all about the difference between diffusion, osmosis, and active transport. This is also important for your exam. Next, there is the osmotic pressure. This is very much important for your exam. Osmotic pressure. Osmotic pressure is the minimum pressure that must be exerted to prevent the passage of the pure solvent into the solution when the two are separated by a semi-permeable membrane. And here is the definition, which is very short and easy to memorize. That is, osmotic pressure of a solution is a measure of its tendency to take in water by osmosis. Now we'll just have a look in this picture, where the application of osmotic pressure is been working here. Due to this osmotic pressure, the piston will be risen up. That is the process of osmotic pressure. Now we will know about tonicity and the process of tonicity. There are three types of tonicity, that is isotonic, hypotonic, and hypertonic. Let's learn about this. Tonicity, relative concentration of the solution that determine the direct and extent of diffusion, is called tonicity. Based on it, the solution can be of three types: isotonic, hypotonic, and hypertonic. We will know its meaning and definition as well, but the most important thing is its application. Isotonic, iso means same, tonus means tension concentration. So the relative concentration of water molecules and the solute on either side of the cell membrane is the same in such a solution. Means in isotonic solution, there is no need movement of the water molecules across the cell membrane. That means there no osmosis will occur, and as there is no osmosis, there no change will occur in isotonic solution. You can see this clearly in this picture. This animal cell has been kept inside this isotonic solution. And regarding this, sorry, this is not the animal cell, plant cell has been kept in this isotonic solution, and as this is kept in isotonic solution, there no change will occur. Now we'll know about hypotonic solution. Hypo means lower. In this condition, the solution outside the cell has a lower solute concentration than the fluids inside the cell. As a result, the water molecules from outside will move into the cell, or endosmosis will occur. And students, I hope you have it in your mind that in case of endosmosis, the cell swells up. So in hypotonic solution, we will see that when this animal cell has been kept in hypotonic solution, this has been swelled up, and this plant cell, when it is kept in hypotonic solution, it will also swell up due to this endosmosis process. Now this is hypertonic solution. Hyper means higher thirst. In this condition, the solution outside the cell has a higher solute concentration than that of the fluids inside the cell. Consequently, the water molecules from the interior of the wall will move out, or exosmosis will occur. And as you know, due to exosmosis, the cell will shrinkage. This is clearly depicted in this picture. This animal cell has been kept in the hypertonic solution, and due to this, the water has comes out of this cell, and for This, as a result, the cell has been shrinkaged, and this plant cell has also been kept in hypertonic solution, and for this the cell will shrinkage after some time because there is exosmosis is taking place. Now, what is the conclusion of these three process? In isotonic solution, cell shape and size will remain unchanged. In hypertonic solution, cell will shrink in size and loses shape. And in hypotonic solution, the cell will slightly enlarged. Now, these three things has been mentioned in this whole picture, and for your help, I have given this all this picture together so that you can understand it better. In isotonic solution, as you can see, there is no change is occurring. In hypotonic solution, the cell is being swelling up because there is endosmosis is occurring, and in hypertonic solution this cell will shrinkage because water will comes out of this cell. So that's all about this three process. Now you need to know about active transport. What is active transport? Active transport is the passage of a substance from its lower to higher concentration through a living cell membrane using energy from the cell. Actually, active transport is in a direction opposite to that of diffusion. What occurs in diffusion? In diffusion, the molecules move from the higher concentration to the lower concentration, and in active transport, the molecules move to that of the lower concentration to the higher concentration area or region. Now, passive transport. Passive transport is nothing different from the diffusion. Passive refers to requiring no input of energy. There is a free movement of molecules from their higher concentration to that of lower concentration. So passive transport is quite alike to that of diffusion, where there is the main difference is that in passive transport there no input of energy will be occurring. Turgidity. This is also important for your exam. Turgidity. Turgidity is the state of a cell in which the cell wall is rigid, means tough, and stretched by an increase in the volume of vacuoles due to the absorption of water. The cell is then said to be turgid. So what is turgidity? Turgidity is the state of a cell in which the cell wall is rigid and stretched by an increase in the volume of vacuoles due to the absorption of water. And here this cell will have to be known as turgid. That means due to the absorption of water, the cell will swell up, and this condition of cell is turgid or turgidity. What do you mean by plasmolysis? Plasmolysis is the concentration of cytoplasm from the cell wall caused due to the withdrawal of water when placed in a hypertonic solution. So plasmolysis means when the cell will be kept in hypertonic solution, then the water will comes out of the cell, and due to this the cytoplasm will be shrinkaged, and for this the plasmolysis will occur. Now flaccidity. Flaccidity is the condition in which the cell content is shrunken and the cell is no more tight. The cell is then said to be flaccid. So flaccidity is the condition in which the cell content is shrunken and the cell is no more tight. This is flaccidity. Now you can see this whole things over here. Due to this is osmotic pressure which is occurring from outside to inside, and turgor pressure is acting from inside to outside on the cell wall, and what wall pressure is being occurring from outside to inside. So this whole picture shows the three things: the osmotic pressure, turgor pressure and wall pressure. You can just have a look for the betterment of understanding. Now this picture clearly depicts a turgid cell itself. You have to memorize this picture for your exam. Next, we'll move to the examples of plasmolysis in our daily life. Salting of meat or addition of salt to the pickles is a method of killing bacteria by plasmolysis. Water is drawn out of the bacterial cell, and as a result the bacteria dies due to plasmolysis. Weeds can be killed in a playground by sprinkling excessive salts around their base. Excessive application of fertilizer in the agricultural fields may similarly damage the roots and diminish the yield as well. So plasmolysis can be effective for killing these bacterial cells, or when salt is added to the meat, then the plasmolysis occurs. So this is the benefit of us, because in this process the bacteria dies. But if we apply much salt or much fertilizer in the agricultural fields, then as a result of the harmful effects of plasmolysis, then the crops will be ruined. You can see this is the germinating seed develops force to push through the upper layers of the soil. So this is occurring here. The germination of seeds has been mentioned in this picture, and due to this, germinating seed develops force to push through the upper layers of the soil. So this seed will move to the upper layer of the soil due to this turgidity. Now, root pressure. Root pressure is the pressure developed in the roots due to continued inward movement of water through cell to cell osmosis, which helps in the ascent of cells upward through the stem. This is the root pressure. Now we need to know the experiment of root pressure. These are the manometers, and the process of root pressure has been mentioned in this picture. You just have to look at this picture and need to understand this root pressure system. Due to this root pressure, this mercury level will rise up after some time when the process will start. And this the process of the turgidity. You can see this mimosa tree which puts down as soon as we touch it. So this occurs due to the turgidity. Here there is turgor pressure is acting. Due to this is now strengthened here, and as soon as we will touch it, it will just move down, because water level will just go down as soon as someone will touch over here. This is like a sensor which acts due to this turgidity or turgid pressure. So you can see here, this is the mimosa pudica. When leaf is being touched, cell of the lower side of the pulvinus lose water, and as a result the petiole collapse and it will move down. So that's all. This picture is very much important for your exam. This is the process of absorption of water, that is root here, and this is the xylem which is moving the water upward. This one the petioles, and that the whole thing is endodermis. These are the cortex, that part is known as epidermis, and these are xylem which are already I told you before. So this is the cross section of a plant. You need to memorize this picture. This is very much important for your exam. If you find any problem, just pause the screen and just have a look on the video once again. Now the guttation. In certain plants like tomato, grass, banana or ferns, the root pressure is high enough to force the water all the way through the stem and comes out through the ends of the leaf veins. This water appears as tiny drops along the margins or the tips of the leaves, especially in the early mornings. As a result, the excessive loss of water occurs, and this process is known as guttation. This guttation occurs in the special plants like tomato, grass, banana or ferns. And this guttation is helpful for creating a force in ascent of sap as well. This is the root pressure which builds up due to osmosis and turgidity of root cells and up into the xylem vessels. You just have a look in this picture. This is not so much important for your exam. Just look at this picture. Here the water is being going upward to the stem from these tissues, these epidermal tissues. Now this experiment shows that xylem tissue of the plant carries water to the upward stems of a plant. And for this experiment we have mixed up some eosin solution into this beaker, where as soon as this plant will absorb water, or as soon as this roots of this plant will absorb water, then this eosin will go upward to the stems and the leaves as well. And if we made a cross section of the leaf or the stems, we will see that this xylem part will be colored red, will be red in color due to this eosin. And as a result we can see that, or we can say that this xylem portion of the plant is helping in the absorption of water, because this xylem portion has only been colored red for this absorption of water. Now we need to know certain notes, that vascular bundles in the stem, root, leaf, stalks and leaf veins are all continuous and form an unbroken system of tubes. Collectively, they form the transport system throughout the entire plant. Water and salt level travel upwards mainly in the xylem, and food substances travel up and down in the phloem part. Now we have finished the chapter. We will just have a look to the whole chapter at a glance. The first point is all plants need water for photosynthesis, transportation and transpiration and mechanical function, which is known as turgidity. Number 2, roots and especially the root hairs provide enormous surface area. Next, minerals are required as constituents of cell and cell organelles. Next point is root hairs contain cell sap of a higher concentration than that of the surrounding water. So the process of absorption of water depends on the process of osmosis. Diffusion. Diffusion is the movement of molecules of a substance from a region of their higher concentration to that of the region of their lower concentration. Osmosis. Osmosis is the diffusion of water molecules from the region of higher water potential to that of lower water potential through a semi-permeable membrane. Next point, osmotic pressure is the pressure by which the water molecules tend to cross the semi-permeable membrane by osmosis. The next point is active transport. Active transport is in a direction opposite to that of the osmosis, as for example from lower to the higher concentration of solutes. The wilting or dropping of leaves is due to the loss of turgidity. Water along with mineral salts absorbed by root is known as sap. The continuous absorption of water by the roots builds up a pressure which is known as root pressure that pushes the sap up through the stem. So that's all about this chapter. We have finished the chapter. I hope students you have understood everything very well. If you have any problem, just let us know in the comment section. And if you have benefited from this video, then share it with your friends and like our videos. Thank you a lot. Have a nice day. Thank you.